Hi, my name is Zach Nagel, and I'm an assistant professor at the Harvard T.H. Chan School of Public Health in the Department of Environmental Health and at the JBL Center for Radiation Sciences. The research in my laboratory is focused on understanding the health effects of environmental exposures, particularly DNA damaging agents like ionizing radiation. DNA damage is dangerous because it can alter the blueprint of the cell, which tells the cell how to behave, contains the key information that regulates how cells work. One of the most familiar and tragic examples of this going wrong is cancer, where alterations in the DNA blueprint cause a cell to proliferate uncontrollably. We know that cancer is caused by DNA damaging agents, and that DNA damaging agents are ubiquitous in the environment, including UV light, radon gas, tobacco smoke, and air pollution, many others. We know that people sustain DNA lesions at a very high rate. In fact, during the time it takes to listen to this video, each of us will sustain trillions of DNA lesions throughout the cells in our body. So why is it that we don't all get cancer? This is the fundamental question that drives research in my laboratory. To study this problem, we are investigating a process called DNA repair, which we think is one of the major reasons why we don't all get cancer, and possibly explains differences in susceptibility to cancer. So to provide an idea of how we do this in my lab, I'd like to introduce one of the projects we're doing. We're collecting blood samples from lung cancer patients and healthy controls and comparing the ability to repair DNA damage using those lymphocytes and a specialized assay that we've developed. The way our assay works is we develop reporters in vitro that can then be introduced into cells and then provide a fluorescent signal that tells us how well the cells repair several different types of DNA damage. So this is our flow cytometer, and this is the instrument that measures fluorescent signal in individual cells. The cells go into a tube that's placed right here, and the sample gets introduced through this injection port, which passes it through a series of tubes and lasers that actually detect different colors at different points along the trajectory through the machine. It's counting cells up to, right now we're seeing about 4,000 cells per second that are passing through the system and being quantified for their fluorescent reporter expression. So this is our high content imaging system, which is an alternative way of measuring fluorescence in individual cells. So what we see on screen are a series of cells that have been circled and analyzed for little dots that represent points of DNA damage that have been detected with an antibody. And this instrument can count up all those dots, which in the past one would have to do by counting by hand. The long-term goal of the research in my laboratory is to translate these basic research findings related to DNA damage and repair to accomplish personalized therapy and personalized prevention of diseases that are associated with DNA damage and inefficient repair of DNA damage.